So, hello. My task today is to start with another interesting topic for you. You have had already you have already three lectures from cytology, and today's lecture and today's topic is to start with general embryology. That means from the beginning some words about this science or this interesting topic. Then we will look on the fertilization and the first 2 weeks of the development of the blastocyst. So embryology is something else than histology. Histology is a science which is explaining the tissues. The tissues, the cell, and later the description and the composition of organs which are composed from tissues. Embryology is a science that is describing or explaining the development of an organism. Embryo is one part of the word, and logos means science is the second part of the word. Logically, we are starting our description or explanation from the fertilization of an oocyte. As you see on the picture, this is the spermatozoa, the sperm, and this is the surface of the oocyte. This layer is zona pellucida, which is surrounding the oocyte. And this description should end logically by the childbirth of the organism or of the human by a natural way or by a caesarean section. That means that embryology is explaining the whole prenatal development of the organism. Pre means before, natus before the birth, up to the birth. After the childbirth is starting the logically postnatal development. The postnatal development begins by the childbirth and logically ends by the death of the organism. We will concentrate logically in our 1 year explaining to you in embryology mainly on the prenatal development. Basic stages. The first 2 weeks. I will look if it's possible to draw some pictures. Yes. From the fertilization, the first 2 weeks. It's so-called the development of the blastocyst. Today, from the zygote, we get to the next stage, which is called the morula. From the morula, I will try to get to the next stage, which is called the blastocyst. And the later blastocyst has to undergo the implantation into the endometrium, in the tunica mucosa of the uterus. So this is the task of today's lecture. The next lecture will start with the third week, and there we have to explain the next very important procedures like gastrulation, notogenesis, and the beginning of the development of some organs in the small body of the embryo. In this moment, it's the embryonic development up to the 8th week, and we call it an embryo. From the 9th week to the childbirth, it's not anymore the embryo, but it's a fetus, please. Fetus, and this part of his development we logically call the fetal development. So, once more, the first 2 weeks blastogenesis, development of the blastocyst after fertilization. The second period is the embryonic development, ending on the end of the 8th week. And from the 9th week till the childbirth is the fetal development. Now let's concentrate on a human, because this is our basic task, of course. So I hope you know that gravidity as the human is lasting around 280 days. These are 40 weeks or 9 calendar months or approximately 10 lunar months. 1 lunar month has 28 days. Times 10 we had our famous number 280. You can even count approximately the date of the childbirth. So here is a simple method or simple help how you can count the date of the childbirth. The obstetrics likes to divide the whole gestation, the whole gravidity of a human into 3 trimesters. The first trimester, the first trimester, weeks from 1 to 16. So logically fertilization, and then is starting the first week. And you see it's nearly to the 8th week, to the end of the embryonic development, and the first part of the fetal period, the first trimester. The second trimester is from the 17th to 28th week. That's very important, this part, because maybe you know or not, but on the 24th week is taken as a very important borderline. Up to the 24th week, if runs out a dead fetus from the body of the mother, it's automatically abortion. After the 24th week, that means from the 25th week later, it's a preterm newborn baby, preterm childbirth. So the 24th week is a very important borderline that till this time it's abortion, after this time it's a preterm newborn baby. Vitality is a question in this moment. Around this period, this time, around this time it's a problem. Why? Because around the 7th month of the fetal development, in the lungs are developing pneumocytes. Pneumocytes, of course, type 2, are starting to produce surfactant. Surfactant is a very important layer lying on the surface of your alveoli, and now it depends how much of surfactant is produced around this time. I don't want to go to details today, but one very important question is the surfactant in this moment, around the 6th, 7th month of the fetal development, and the amount of surfactant on the surface of your alveoli in your lungs. The third trimester is very clear. It's very clear from the 29th till the 40th week, and there is a clear fetal development. Around the 40th week, a normal childbirth, and you have a newborn baby in this moment. I hope that you could remember one very important name in your life, and this is Professor Jan Evangelista Jiras. This professor was many years working in embryology, many years on gynecology and obstetric clinics, and he was also working on the second medical faculty here in Motel. I think he has come with very good ideas during his professional life, and of course also famous Jiras 6 stages of human prenatal development. You see that he has given some letters, basic letters and numbers to his stages. So as you see, our similar borderline, my lecture is ending in this moment from his A1, A2, A3 and 4 stages. And you see that from the third week, we'll run on the next stages, and this will be the topic of the next lecture. So please, he was a very famous embryologist and gynecologist, and a very good professor in obstetric. So, little bit from history. You could know some, maybe you know, or you could gain from this lecture some new important names. The first very important, and maybe it's the right year, I am not sure completely, but they say it is so, 1677. These two gentlemen, Anton van Leeuwenhoek and Johann Hahn von Arnheim, from Holland, Netherlands, Holland. Anton van Leeuwenhoek has also built a very good microscope on this time. Not a good one, but a more precise microscope. Those two gentlemen has described for the first time spermatozoa. They have seen it in the simple microscope, but they did not know nothing about the function, of course, in this time. Mr. de Graaf has seen in the near the same years and described in the ovary of a dog follicles. Because also a dog, a female dog, has also follicles in the ovary, developing, growing follicles. But he did not understand that there is an oocyte or something like this. Many years later, von Baer, this gentleman, described an oocyte inside a follicle in the ovary. But of course, he did not know why it's here, for what it's here, any idea of the function. In those years, those three important gentlemen has given to the whole world, Schleiden, Schwann, and Mr. Purkinje has given the cell theory. That the body, the human body is built, and the body of organisms are built from cells. So of course a cell is an oocyte in this moment, and a very interesting cell is a spermatozoa. Another important date was year 1978, when was born Louise Brown, very nice small girl, by in vitro fertilization. In vitro fertilization. The authors are Mr. Edwards and Mr. Stepsel of this in vitro fertilization. Little bit the history as you see in Czech Republic. I think it was clear that for many years and centuries the obstetric assistants, midwives, were the most important helpers during childbirth. Then for many next centuries, obstetric was a part of surgery medicine. It was not a separate clinical subject. This famous Latin few words means that this art is not for men. That means only for ladies. So you see that the midwives and the assistants, ladies, were very important in the childbirth of many people during many centuries. And then started the first lectures for medical students in Prague around the year 1840. Then was built a first very small obstetric clinic in Prague, in the Charles place, there in today's hospital, where is the place of the first medical faculty. And then was built a larger obstetric department in the Apolinarska Street from year 1875. It became a very nice obstetric medicine, a very nice separated subject. So little bit of history that you will be more clever. So today I said already many times a neck, oocyte. We can divide some types, or we know some types of oocytes according to some basic lines. The first important line is that a cell has got, an oocyte has got a cell membrane, an oocyte has got the nucleus, the oocyte has got the cytoplasm. In the cytoplasm organelles, but as well in the cytoplasm, like other cells, has got cell inclusions. You know that cell inclusions we take as proteins, saccharides, polysaccharides, lipids. We can combine glycoproteins, we can combine glycolipids, everything is possible. And now it depends how big quantum of the cell inclusions are in the cytoplasm of the oocyte. A Greek name, lekitos, means in English yolk. So logically, now we can use lekitos in all types of those oocytes, and only the first part of the longer name means how much of these cell inclusions are present. So logically, the first very small oocytes with the abbreviation A, let's settle. You see that they are very small, 20 to 40 micrometers. In some sea mushrooms, yes, let's settle oocytes. This A means that there is also little minimum amount of cell inclusions. It's not completely empty. So let's settle oocytes are the smallest one with a little amount of those inclusions. Next name, let's go to a bigger oocyte, oligo. Oligo means little, small, oligo. Lekitos, yolk. That means oocyte with a little amount of cell inclusions. You see that we are going in micrometers to a higher number. You can see them in the microscope, in the light microscope, such oocytes. And in the cytoplasm is a small amount of glycogen, lipid droplets, glycoproteins, and other cell inclusions. A big group of chordaceae, this name is thanks to Chorda dorsalis, which is developing in those organisms. In English, notochord, Chorda dorsalis. And to this big group belongs 3 smaller groups: tunics, lancelet, and vertebrate. Of course, human, we have vertebrae, we belongs to mammals, and a human belongs to mammals with a placenta, please, with a placenta. Tunics, hops and salmons, they are living in the sea. In their small bodies is developing the notochord, but not staying the whole life. Branchiostoma lanceolatum, please, is a small, very small fish living in the sea, hidden in the sand on the bottom of the sea, coming out sometimes during the night, day, and running back into the sand and hidden there. This Branchiostoma lanceolatum, lancelet, has the notochord for the whole life. And of course, human, a mammal with the placenta. During development, from the 3rd week, is developing Chorda dorsalis in the body of the embryo, yes, but it's vanishing because it's replaced by vertebrae, column from vertebrae, and there is a small remembrance that we have had this notochord in our body during the development. It's a part, I don't want to go in details, but it's a part of the intervertebral discs between the vertebrae. And so looks the lancelet. Branchiostoma lanceolatum or Amphioxus lanceolatus. It's a very small and such a very thin sea fish. In the body, the notochord has got the whole life. The basic axis structure in this body of the small sea fish, so has got the lancelet. Types of oocyte, let's move to bigger. You see a new name on the beginning, meso. Meso, something between, meso, middle. Lekitos, yolk. Those oocytes you can see already with your eye, you don't need a microscope. So, some types of fishes. Maybe you know the names, lamprey, beluga, lungfish general. So some types of fishes, and of course all amphibians has got this type of oocyte, mesolexital oocytes. And now the last group, from meso, we have to move to a big amount of yolk, and we are at polylexital oocytes. Polylexital oocytes. So they are already macroscopically seen from millimeters up to, maybe you have seen somewhere in a film an oocyte of a Tyrannosaurus rex, for example. So maybe around 80 centimeters could be his oocyte large. With bone skeleton fishes, all sharks and rays, all reptiles, birds and primeval reptiles has got polylexital oocytes. And one interesting group from mammals, and polylexital oocytes as mammals which are laying eggs. They haven't got a placenta. So maybe you know the Ornithorhynchus anatinus, duckbill platypus. Maybe you know some marsupials. Maybe you know kangaroos. Yes, so all those and still other examples of mammals laying eggs. So they have polylexital oocytes, please. So this is one possible line how we can divide oocytes according to the amount of the yolk. Second possible line is, where is the yolk present? Where is the yolk present in the cytoplasm? The distribution of the yolk. Yes. So the first group is iso. Iso means everywhere. The inclusions are really around the nucleus. The nucleus is in the center of the oocyte, in the center. And around this nucleus, in the cytoplasm, you could find and you could prove the cell inclusions. Logically, all oligolexital oocytes are isolexital. Telo means telo on one side, on one pole. So this logically polylexital and meso and poly. Those big oocytes on one pole has got the accumulation of the yolk. This is the vegetative pole of the oocyte. And on the opposite pole, which we call the animal pole, so here will be running the cleavage. Here will be running mitotic divisions of the cells. But the nutrition those cells are getting for some time from the vegetative pole. So this is now the second group, and this is not a big group. Centro means in the center. The yolk is in the center of the oocyte, the nucleus is on the side, so you can find them in some index. Such interesting centrolexital oocytes. And the last line and last possible division to see different oocytes according to the cleavage. That means the zygote, how will be dividing? If the zygote is completely dividing into two daughter cells, into complete new two daughter cells, then such oocytes are holoblastic. The whole zygote is dividing into daughter cells. And meroblastic oocytes, logically, the cleavage is running only on the animal pole, not where is the yolk. So you can see new cells only on the animal pole of such an oocyte. So logically, polylexital eggs are meroblastic. So please, I think you could remember, because we are here Department of Histology and Embryology of the Medical Faculty for Humans, I think you could quite nicely remember the oocyte of a human. This is quite enough. A human oocyte is oligolexital, isolexital and holoblastic. Total cleavage. Cleavage at a human is in the time asynchronous. You will see, I will explain it in few minutes. So you see a nice oocyte with a cell membrane, with a nucleus, cytoplasm, where are the cell inclusions distributed everywhere. Iso, let's say, iso. Here you see something like zona pellucida. This is a glycoprotein layer which is surrounding the oocyte. And you see the first layer of follicular cells, which are still around the oocyte. This first layer of follicular cells is called corona radiata. That's a name for a typical first layer of follicular cells surrounding the oocyte. In the moment of ovulation, the oocyte is around 150 micrometers average. 150 micrometers average. So once more, for the last time, I want to leave the oocyte oligolecycal, little amount of cell inclusions. Iso, they are everywhere in the cytoplasm, not on one pole. Holoblastic, when it will be a fertilized oocyte, a zygote, it will divide completely into two daughter cells, completely. So, I think you understood it, and we can move to a next chapter. Gametogenesis, development of gametes. That means of an oocyte and of a spermatozoa. Development of this. In detail, we will have it once more in the summer semester, when we are at the male reproductive system in the testis, and in detail once more when we are in the female reproductive system in the ovary. The spermatozoa are developing in the testis, in the seminiferous tubules. Oocytes are developing in the ovaries, in growing and developing follicles. There is a big difference between the development of spermatozoa, spermatogenesis, and of an oocyte, oogenesis. Big difference in three basic differences, in three basic points. To the right number of chromosomes, you need not a mitotic division, but a meiotic division. Meiosis, please. This is important. Mitotic division, nothing strange, you know it. When the cell is dividing by mitotic division, the result, two daughter cells, in each cell 46 and 46 chromosomes. In each daughter cell. Once more, 2n. 46 and 46 chromosomes. By a meiotic division, we have to come to the result that in the last cell will be only 23 chromosomes, each chromosome with only one chromatid. This is the first basic task of meiotic division, to reduce from diploid number to haploid numbers of chromosomes. Second important thing during meiotic division is happening in the prophase of the first meiotic division, that in the packaging stage or phase is happening something very interesting, what is called crossing over. The second very important thing during meiotic division. Meiotic division is composed from two divisions. The first meiotic division happens, and the result are two cells, in each cell 23 chromosomes, yes, but each chromosome has still two chromatids. A very short interface, and this new cell will be dividing once more into next two cells, and these two cells will have 23 chromosomes, but each chromosome only one chromatid. Haploid number. Of course, when a spermatozoa with 23 chromosomes meets an oocyte with 23 chromosomes, the new cell, zygote, has once more 46 chromosomes. So, the development of spermatozoa is happening in the seminiferous tubules in the testes. This long procedure, or maybe long, maybe short, it depends on the temperature. You know that the testes are sitting in the scrotum for one very important reason. The optimum temperature for the development of spermatozoa is 35 to 36 degrees of Celsius. That's the reason why the testes has to go from the body cavity into the scrotum during the fetal development. And the first part of spermatogenesis are cell divisions, including, of course, meiotic division, meiosis, of course, meiotic division. The last stage after the second meiotic division is a very small cell, a spermatid. This spermatid has 23 chromosomes, each chromosome one chromatid. It cannot divide anymore. These nice, very small cells will now undergo spermatohistogenesis, differentiation from a spermatid into a ready, nice, definitive sperm. The whole procedure, if you are interested, from the first meiotic division, from the prophase of the first meiotic division, till the end, when it's a ready sperm, is lasting 64 to 68 days, but under this ideal and optimum temperature 35 to 36 degrees of Celsius. The first cells from one spermatogonia, where will start the prophase of the first meiotic division. From one spermatogonia will be on the end 4 spermatids. From 4 spermatids, 4 spermatozoa. 23 chromosomes. One of the 23 chromosomes in 2 will be chromosome X. In the next 2 will be chromosome Y. Here you see a nice spermatid on the beginning, this cell. And now you see slowly in this line running down here the procedure of spermatohistogenesis to the result a definitive spermatozoa. You see that only the procedure of spermatohistogenesis is lasting around 24 days. 24 days. And you see how the cell, which is round or slightly oval shaped, with a spherical nucleus, this is a spherical nucleus, is changing and differentiating slowly and steadily in the right shape of the spermatozoa. First, look on the nucleus. The nucleus from the spherical shape is changing into a very thin shape, as you see here. In this head of the spermatozoa are sitting those 23 chromosomes. How is it possible that the shape can change so nicely, easily? It's thanks that some important proteins in the nucleus, which are normally in each cell histones, are degraded and replaced by much smaller proteins, protamines. This dark line is DNA. And thanks to protamines, it's possible that the nucleus can, in this interesting procedure, change the shape. Thanks to new proteins, protamines. The Golgi, you see here the Golgi apparatus. Yes, the Golgi apparatus is starting to be accumulated on the surface of the head, forming the acrosomal cap. Acrosomal cap with many interesting hydrolytic enzymes in the acrosomal cap. Then is the neck. This is somewhere this part, neck, and then is the middle piece of the spermatozoa. And what is interesting here, this mitochondrial sheet, one mitochondria to another. The big factory for ATP molecules, adenosine triphosphate molecules, energy for the movement of the tail. The tail has this principal part, you see only a small part here, and the terminal part of the tail. The whole spermatozoa from the head till the end of the tail is long 60 to 65 micrometers. 60 to 65 micrometers. So you see once more, from one spermatogonia, the right result are 4 mature sperms. Of course, as you remember, 2 has got chromosome X and 2 has got Y. 1 from the 23 chromosomes. Of course, if this spermatozoa with X will meet the oocyte, has only X chromosome, it will be a girl. If this spermatozoa with Y chromosome will meet the oocyte with X chromosome, it will be a boy. So I hope this is clear. And few words about the development of the oocyte, because there is a big difference against the spermatozoa. Oogenesis is something completely different than spermatogenesis. It is different in 2 basic lines. In 2 basic lines it's very different. The first big difference, still during the fetal development, the oocyte is starting the prophase of the first meiotic division, still in the uterus of the mother, the small girl, fetus, fetal development. And the prophase will run leptoten stage, zygoten stage, pachyten stage will happen with crossing over, and the next stage is called diploten. And now Mother Nature is stopping the whole division in the diploten stage, of the prophase of the first meiotic division. This period, this stop, can last in some oocytes till the puberty, till 13 years. In some oocytes, even 52 years can last this stopped period in diploten stage. Before ovulation, the oocyte will finish the first meiotic division, and one cell is falling away, which we call the first polar body, and of course the oocyte, the secondary oocyte is here. In the first polar body are 23 chromosomes, each chromosome 2 chromatids. In this oocyte, remaining 23 chromosomes, each chromosome 2 chromatids. Few hours before the ovulation will start the second meiotic division. And will come a second stop. Mother Nature is stopping the second meiotic division in the metaphase of the second meiotic division. That means in the moment of the ovulation, rupture of the thin wall of the Graafian follicle. This is ovulation. The rupture of the thin wall of the Graafian follicle. In this moment, the oocyte is in the metaphase of the second meiotic division. If happens the fertilization, the oocyte will finish the second meiotic division. If does not happen the fertilization, any fertilization, the oocyte will never come to the end. It will remain in the metaphase and later, let's say very simply, eaten by the macrophages in the uterine tube. If happens the fertilization, you see after the second meiotic division is falling away the second polar body. As you see, the author decided still to divide the first polar body, it's possible sometimes, yes, into two smaller polar bodies. But nevertheless, in each polar body, 23 chromosomes. Each chromosome has only one chromatid. In this oocyte, which undergoes the second meiotic division to the end, will remain 23 chromosomes, each chromosome one chromatid. So you see, this is the first big difference, second big difference against or to spermatogenesis. Only one oocyte is the result, only one oocyte. You know spermatogenesis, 4 mature sperms. Here only one oocyte is the result. Or 3 polar bodies or 2 polar bodies will vanish. No function. Completely nothing. One oocyte is the result. Second interesting differentiation is, or difference, development of the spermatozoa, 64 to 68 days, it's ready. And here, it stopped in the diplotent stage for 13 up to 50, 52, 55 years. And once more stopped in the metaphase of the second meiotic division in the moment of the ovulation. So you see here are big differences between spermatogenesis and oogenesis. So I hope it's clear. And let's move to the next chapter, next topic, fertilization of an oocyte. Here you see a little nice scheme of the female reproductive organs. So let's start with the ovary, the uterus, the uterine tube, the vagina, the cervix of the uterus. Fertilization, please, at a human. Now let's speak about the human. Is happening at this place. At this place, on this side, somewhere here. The ampullary part of the uterine tube. Ampullary part, or in the ampulla of the uterine tube. The uterine tube is a tube at a human long, around 8-9 centimeters, with 4 basic parts. This part in the wall of the uterus is pars uterina. This green is isthmus tubae uterinae, and the yellow is the ampullary part, ampulla of the uterine tube. This is the infundibulum. Here are such long processes which are called fimbriae. You see them nicely here, fimbriae, which are jumping on the surface of the ovary, catching the fluid, the oocyte, and zona pellucida and follicular cells that should jump after ovulation into the ampullary part of the uterine tube. So, ovulation, rupture of the Graafian follicle, and the oocyte with the fluid, with the zona pellucida and corona radiata. You remember the first layer of follicular cells around zona pellucida are coming into the ampullary part of the uterus and sitting here and waiting for the spermatozoa. Possibility around 24 hours for fertilization. Spermatozoa from the vagina has to move through the cervix of the uterus. The composition, viscosity of the secret of the glands is now optimum for the spermatozoa in this place. So the spermatozoa can move quickly through the cervix of the uterus. Now they have to move through the cavity of the uterus, but half runs to the left, half of them number runs to the right. And now is waiting them a long way, few centimeters in the uterine tube, through the isthmus to the ampullary part to find the oocyte. Please, from this point till this point, for the small spermatozoa, it's around 13, 14, somewhere 15 centimeters. Such a long distance. It's a long distance for a sperm which is long 60 to 65 micrometers. Has to, let's say very simply, has to swim, has to get through a distance around 14 centimeters to find the oocyte in the ampullary part of the uterine tube. Millions of spermatozoa ending in the vagina, thanks to acid pH, millions of spermatozoa ending here. But the quickest are coming through the cervix and now the uterine tube. Logically, you use thousands and thousands and thousands because some are moving to the opposite side where it's not the oocyte. And then still the long way, 8 centimeters through the uterine tube. Many are ending on the way here because there are also present macrophages. And of course, macrophages and a sperm is a different cell. It's not a cell from the body of the lady. So macrophages and phagocytosis is running in the uterine tube. So not all from the vagina can get to the ampullary part of the uterine tube. So in the ejaculate, I think it's such a normal optimum volume is 4 to 6 milliliters at a healthy man. In this 4 to 6 milliliters would be nice to have the right number of spermatozoa. 200 million to 400 million of spermatozoa. This is a really optimum number. You remember I said millions of spermatozoa ending their life in the vagina, acid pH. They will give you, you can gain also a spermiogram or spermatogram, and they will explain you and they will write you on a paper the result. Oligo, you see oligo, little amount of spermatozoa. Azo, they are not moving. And now are different names, what is happening to spermatozoa. Don't forget, from this point till the end of the tail, 60 to 65 micrometers long cell. In the head, 23 chromosomes, each chromosome one chromatid. Here are the mitochondria accumulated. So, what is interesting on this? The ejaculate is is the secret as a result of some glands, prostatic glands, seminal vesicles, the testicular fluid from ductus epididymitis and testis with many spermatozoa. The ejaculate has a very interesting composition, which is helping the spermatozoa to survive in the vagina and to move on. Many interesting enzymes, sugar, fructose, for example. Very important thing which has to undergo the spermatozoa in the way to the oocyte. Capacitation is starting in the uterus, but mainly running in the uterine tube. Because in this long uterine tube, around 8 centimeters, the spermatozoa are swimming till the oocyte around 8 hours. So mainly in the uterine tube is happening this. You see that these are several important points what happens on the membrane of the acrosomal cap. If the capacitation would not happen, the enzymes in the acrosomal cap could not be released to help the head how to get through the zona pellucida. So those basic points are happening during capacitation. Glycocalyx is released. Glycocalyx, I hope you know that these are short chains of oligosaccharides, glycolipids, glycoproteins on the surface of the cell membrane. Glycocalyx. New distribution of phospholipids and oligosaccharides on the membrane. So this is the first important thing which is happening in the uterine tube during the way of the spermatozoa to the oocyte. To the oocyte will get from 400 millions of spermatozoa will get maximum number 250, 300, sometimes 400, maximum 500. 500 from 400 millions, maximum 500 spermatozoa. As you see, the whole fertilization is very simple in fact. We have to connect two nuclei. Each nuclei has 23 chromosomes, each chromosome one chromatid. Once more, the new cell, the new zygote has 46 chromosomes. 46. So both the optimum number is the optimum fertilized, so it's called the possibility for fertilization is around 24 hours. Capacitation of the spermatozoa I explained. Now the spermatozoa is getting to the oocyte. In the ampullary part of the uterine tube will happen the acrosomal reaction. The head will move through zona pellucida to the membrane of the oocyte. Has to happen the contact and the fusion of the membrane of the sperm and oocyte. What is inside the sperm will get into the cytoplasm of the oocyte. The last very important part is the cortical reaction. This is the problem not of the spermatozoa but of the oocyte to prevent polyspermy. Acrosomal reaction. You have to release all important enzymes. All important enzymes in the acrosomal cap. The most important is acrosin. Those enzymes are working on proteins and glycoproteins in zona pellucida to prepare a nice way through zona pellucida, which is at a human 13 to 14 micrometers, to prepare the way or the highway for the head which is moving through zona pellucida to the membrane of the oocyte. So that's the function of those enzymes in the acrosomal cap. Acrosomal reaction. Acrosomal reaction. The most basic protein is ZP3, and now they are really working on the ZP3, those enzymes, forming a nice way for the head to get to the membrane of the oocyte. You see zona pellucida, this is the red. You see the membrane of the oocyte. You see the acrosomal cap, and here are hidden the enzymes in the acrosomal cap. Here are 23 chromosomes. You see how the membrane is vanishing and the enzymes working on the zona pellucida, on the proteins in zona pellucida. And the head slowly and steadily moving through zona pellucida, and it will attach and it will touch the membrane of the oocyte. Very nice picture, but with one mistake. One mistake. Here it's better. When the head is coming into the contact with the membrane of the oocyte, It's never in this position. It's always in an oblique position. So not 90 degrees. Not 90 degrees. But always in an oblique position, please. It's touching the head and the membrane of the head, the membrane of the oocyte. You see here zona pellucida. You see here follicular cells, corona radiata, the first layer of follicular cells around zona pellucida. So here this scheme is already a little bit on. Fusion of these two membranes, and the whole contents of the sperm runs into the cytoplasm of the oocyte. There are two basic and important proteins. Maybe you have heard about them. Those proteins are very important receptors. One such a protein is in the membrane of the sperm, isomo, and the second important receptor is in the membrane of the oocyte, which is called UNO. And these two receptors are very good friends. When they meet, they can come and they undergo the perfect contact between the sperm and the membrane of the oocyte. These two interesting names, if you want to know how the science workers came to this. Izumo is from a Japanese name, Japanese word. Izumo means a Japanese shrimp, which is dedicated to marriage. In French, mariage, to marriage. Juno is from mythology, Juno, and it's a god, a lady god, from a Roman god for fertility and marriage. So these two names, one is from Japan and second is from mythology. It was a name of a god, Juno. So very interesting two proteins working as receptors. Isomo is the receptor in the spermatozoa, Juno is the receptor in the oocyte. So the whole contents of the spermatozoa, as you see this, runs things into the cytoplasm of the oocyte. 23 chromosomes in the head, neck, the middle piece with all mitochondria, microtubules from the tail. The membrane of the tail will fall away. The membrane of the tail will fall away from zona pellucida. But the whole contents of the sperm is now in the cytoplasm of the oocyte. The oocyte is in the second meiotic division, you see, in the metaphase. You see the mitotic spindle, you see the chromosomes. The author decided to divide also the first polar body into two. You remember, I said it, sometimes it's possible. Now will happen the next stages of the second meiotic division. Anaphase, telophase. The metaphase will be completed and still anaphase and telophase. The second polar body will be somewhere at this place, second polar body. In the oocyte will remain 23 chromosomes, each chromosome one chromatid. The sperm has brought 23 chromosomes, each chromosome one chromatid. Two things interesting here. The mitochondria. All mitochondria brought by the spermatozoa from the middle piece will vanish. They are signed by a protein which is called ubiquitin, and such old and not functional perfect mitochondria will be dismissed by proteasomes. Proteasomes are structures to break down proteins, polypeptide chains. The proteasomes are present in the cytoplasm of the oocyte, and all mitochondria from the sperm will vanish. In the oocyte are remaining only mitochondria from the oocyte. That means the maternal mitochondria. This is the first interesting thing. And second interesting thing, during the first meiotic division, the oocyte is losing centrioles. They are away, any centrioles. So this mitotic spindle, the microtubules of the mitotic spindle are not built from a centriole, like normally, but only from the pericentral material. Only. They are missing centrioles. So now this fertilized oocyte can be a little bit happy that the spermatozoa is bringing two centrioles. That the spermatozoa has brought two centrioles, distal and proximal two centrioles. Because the zygote will now need centrioles for the first mitotic division. For the first mitotic division. In the cytoplasm of the oocyte, fertilized will form two nuclei, two small nuclei. One is called pronucleus femininus. There will be the 23 chromosomes after the second meiotic division. And 23 chromosomes will be formed in a small, very small round nucleus, which is called pronucleus masculinus. In this small pronucleus will be the 23 chromosomes which has brought the spermatozoa into the oocyte. And the last, cortical reaction. Please, cortical reaction is the problem of the oocyte, not of the spermatozoa. You don't want another spermatozoa in the cytoplasm. This would be next new 23 chromosomes. This would be very bad, I think. So the cortical reaction is very simple. The first fast block is very quick, and you see on this picture cortical granules with some enzymes, and you see the cortical granules are very near the membrane of the oocyte, very near. And when this first spermatozoa will touch the membrane of the oocyte, immediately the contents of the granules are released into zona pellucida, immediately. It's lasting a few times, so the first block is very quick to change the potential membrane, 2-3 seconds. So, but it's enough time, few minutes, that can start the slower part. Cortical granules fuse with the membrane, and the contents is released into zona pellucida. Those enzymes and polysaccharides are changing the chemical composition of zona pellucida, and this will not give the possibility, the chance to a next spermatozoa to move through zona pellucida ahead. This is one very interesting thing which happens. And the second interesting thing, what was found in the last years, after 1 hour after fertilization, will completely disappear this protein from the surface of the oocyte. Will disappear completely. Approximately 50 minutes, 1 hour after fertilization. So this is a third such as block that a new sperm cannot attach to the membrane of the oocyte. So, capacitation of the spermatozoa, acrosomal reaction, contact and fusion of two membranes, cortical reaction, please. Such 4 basic phases of the whole procedure, fertilization. In the ampullary part of the uterine tube at a human, please. So, what we see on this scheme. So you see in the metaphase of the second meiotic division. But he is not a centriole, Also dividing the first polar body, okay. The contents is inside. After the second meiotic division, the first nucleus is seen. Two nuclei, pronucleus of the lady, of the oocyte, pronucleus femininus, pronucleus of the male, masculinus. At this moment, this cell we call an oocyte. When you see these two pronuclei, it's not yet a zygote, it's an oocyte. Then these 23 and 23 chromosomes will completely mix in one big mixture. It's not here on the picture anymore. But imagine that these 23 and 23 chromosomes will mix in such an interesting mess. In this moment, the cell can be called a zygote. Because here you see the first mitotic spindle, thanks to centrioles, of the first mitotic division of the zygote, which is seen around 24-25 hours after fertilization. You see the metaphase, you see here the anaphase of the mitotic division, you see telophase is starting, and the zygote will divide into two daughter cells completely. It's a holoblastic oocyte. Will divide completely into two daughter cells, which are called blastomeres. You see one polar body, second polar body, zona pellucida, and you see one blastomere, second blastomere. Result of the first mitotic division of the zygote. Zona pellucida, oocyte. Why? Because I see the pronucleus masculinus and pronucleus femininus. So it's an oocyte. Then happens a mixture of 46 chromosomes, and you see perfectly two blastomeres after the first mitotic division. So the cleavage started. Cleavage, repeating mitotic divisions, one after another. In this moment, if you would take either this blastomere or the second one, you separate them from both on this level. From both can be completely a new human. Completely new human. This sometimes happens. Sometimes happens such a procedure, interesting, in the uterine tube. And then you have a first possibility how to come to monozygotic twins. So, I hope you understood that in a zygote are once more 46 chromosomes, 23 and 23. Replication of DNA. After 24-25 hours, we see the first division into two blastomeres. One blastomere will start dividing, the second as well. Suddenly we have 4 blastomeres. Next division of blastomeres, we can have 7, 8, 9 blastomeres, maybe 8 here. Please, in time is asynchronous at a human. Not automatically 2, 4, 8, 16, 32, 64. You can see even 2 blastomeres, 4 blastomeres, 7 blastomeres, 11, 16, 18 blastomeres, 21 blastomeres. One blastomere is dividing quicker, second slower. So please, in time it's asynchronous at a human. This dividing zygote is slowly moving through the uterine tube to the uterus. Around the 3rd day after fertilization, we are on a level, look, it's a little bit complicated to count, but I think on a level approximately about 16 blastomeres. You don't see behind. So approximately 16 blastomers. In this moment, we have a new structure which is called the morula. Third day after fertilization is the morula. Zona pellucida is still around. It's holding this compound structure together. That means the new blastomers has to be smaller and smaller and smaller because zona pellucida is still around. They cannot become as large as was the zygote on the beginning. So you see smaller and smaller blastomers by each next mitotic division. You see here 2 blastomers and the polar body. This is zona pellucida. You see 4, maybe 1, 2, 3, 4, 5, 8 blastomers. Zona pellucida is around. And now happens a new important thing, some different cell junctions between cells in the morula. This procedure is called compaction, and we can somehow see 2 basic parts or layers in the morula. The outer part, the outer cells which we call the outer cell mass. Between those cells happens very tight junctions, zonulae occludentes. And logically, in the center of the morula, the blastomers, forming the inner cell mass, there will be only such communicating junctions, something like nexuses, gap junctions. So this procedure is happening now in the morula around the third and a half day after fertilization. Don't forget we are in the uterine tube, in the isthmus of the uterine tube. For repeating, nice morula. Morula, zona pellucida, around, outer cell mass, inner cell mass. Outer cell mass and inner cell mass. And let's move slowly to the next stage, because fluid coming in through zona pellucida from the uterine tube is starting to fill the spaces between the inner cell mass, and the spaces are connecting into a bigger space full of fluid. And in this moment, when you see a space somewhere inside, we cannot call it anymore a morula, but a early blastocyst. You see that the fluid is more and more and more. So this is the blastocell or the cavity of the blastocyst, This outer we will now call trophoblast, and this inner accumulation of blastomers we will call embryoblast. So the inner cell mass, the inner, will form the embryoblast. The outer cell mass will form the trophoblast. Please, when I said early, automatically zona pellucida is still around. Zona pellucida is still around the early blastocyst. Day 3, morula in the isthmus. Day 3. Pars uterina of the uterine tube. Day 4. An early blastocyst is falling into the uterine cavity. And now, day 5. You have to get rid of zona pellucida. You have to get rid of this. If the zona pellucida would be more and more and longer time around, implantation will never happen. So you have to get rid of zona pellucida at the day 5. If this happens, not an early blastocyst but a later blastocyst can attach day 6 to the epithelium of the endometrium and will start the implantation into the endometrium. Day 7, 8, 9, 10, 11, 12. Implantation into the endometrium. So you see fertilization somewhere at this part. Morula at this part of the uterine tube. Starting implantation 6th day of the blastocyst into the endometrium in the uterus. So, this was such a first chapter, and now a few points that maybe you are interested to get more information about the in vitro fertilization. You know that the first nice girl, Louise Brown, was born by this possibility on the 25th of July 1978. Her mother had a big problem with the uterine tubes. Both uterine tubes were completely closed. Logically, the spermatozoa could not get through to the ampullary part, and logically, the morula and blastocyst could not get to the uterus. In Czech Republic, it's also running this possibility how to help young people to get a child. So sometimes it's not successful, sometimes yes. There are only please 3 possibilities, nothing more, 3 possibilities how to help. The first possibility, you have to be sure that the lumen of the uterine tubes are opened. This is important. Then you can try directly into the uterine tube to transform or transfer spermatozoa. Gamete intrafallopian transfer. Spermatozoa inside the uterine tube to transfer. That means the name, gift. If this happens, many spermatozoa in the uterine tube happens here in the ampullary part fertilization, and the morula and the blastocyst moving through the uterine tube to the uterus. Second problem, embryo transfer. Really the uterine tubes are closed. It's sure that the morula and the blastocyst would not move to the uterus through the uterine tube. Okay. They gain the oocyte from the lady just before the ovulation. They know the menstrual cycle of the lady, and by a laparoscope they try to gain the oocyte. This happens from the Graafian follicle. From the Graafian follicle, before it ruptures, they gain the oocyte. Then they give it into a cultivation medium and add thousands of spermatozoa. Fertilization happens here in this special medium. They are watching it, they are controlling it each day, and they wait. Now the cultivation runs 2 to 6 days. That means what I said, on the 6th day, a later blastocyst, zona pellucida, is available. At this stage, they take out this blastocyst and transfer it back into the uterus to the epithelium of the endometrium, and implantation will run now of this blastocyst into the endometrium. It's more successful to take 2 blastocysts. How? No, logically. After the first mitotic division, they take 1 blastomer, second blastomer, they separate them, and they have 2 blastocysts. So it's better to give back into the uterus, not 1 blastocyst, but 2 blastocysts. The uterine tubes are completely closed, so they have to try this possibility. In some countries it's possible to give the blastocyst into another lady. Yes, the law allows it. In Czech Republic it's forbidden, please. Back into the lady from which was the oocyte. Last possibility, intracytoplasmic sperm injection, ICSE. Microinjection, one spermatozoa you push into the oocyte. One, you push into the oocyte. Zygote is given back into the uterine tube of course, to the ampullary part of the uterine tube. You see zygote, transfer, zygote, transfer. And the uterine tube has to be opened, because logically will happen the cleavage, morula, early blastocyst, and this is moving to the uterus. So there are only three possibilities how to help by in vitro fertilization today. So I was explaining this. You see nice two pictures. Once more, zona pellucida is around blastocell or cavity of the blastocyst, trophoblast, embryoblast. And we have on the picture, on this nice picture, an early blastocyst, because zona pellucida is still around. Scheme of a early, later blastocyst. You see zona pellucida is not around anymore. Embryoblast we see on a pole which is called the embryonic pole. The opposite is up embryonic pole. Cavity of the blastocyst is present, of course. Cavity of the blastocyst, early, later is present. How to get rid of zona pellucida? This interesting procedure is called hatching. I will show it on a picture, and here you see it, hatching. You see that this line is zona pellucida, and the blastocyst hatches out from the zona pellucida, and this was before an early blastocyst, and now in this moment it's a later blastocyst. Here is a cavity of the blastocyst. You see the embryoblast and you see the trophoblast cells. So this is an interesting procedure which is called hatching, and it hatches out thanks to some enzymes which releases ZP proteins in zona pellucida, and the blastocyst jumps out, this is lasting few seconds, jumps out from zona pellucida. In the moment it's a bigger blastocyst, an increase in the size, in the volume, and on the world you have a later blastocyst. Implantation would never happen if zona pellucida would be remaining on the blastocyst. And you see the 6th day. This is the endometrium of the uterus, and how the embryonic pole attaches to the surface epithelium of the endometrium. Simple columnar epithelium. Please, this is simple columnar epithelium. Simple columnar epithelium on the surface of the endometrium, and to these epithelial cells attaches always with the embryonic pole the later blastocyst. So this is clear 6th day, and this is the 7th day after fertilization. So attaching to the surface epithelium. 7th day is starting the implantation into the endometrium. This whole blastocyst has to get inside into the endometrium. There are special receptors on the membrane of the epithelial cells. Once more, oligosaccharides like those receptors, and it always attaches with the embryonic pole, always with the embryonic pole. You see the 6th day on this scheme and the 7th day. So I think you understand what is the surface epithelium of the endometrium, embryonic pole, embryoblast, trophoblast, blastocyst cavity. And now something new, just starting from the 7th day. Those cells of trophoblast, they are starting at this moment, at this place, and there is the endometrium on this pole. Intensive mitotic divisions. New and new mitotic figures we see. Metaphase, anaphase, telophase in the cells. And the cells are intensively dividing. another daughter cell will go into a next mitotic division, and the next daughter cell will fuse with the neighbor, and they are starting to form this finger-like structure. You see still the nuclei inside, but not the borderlines of the cells anymore. And they form this new structure, which is called, it's written down here, syncytiotrophoblast. Syncytiotrophoblast. Syncytiotrophoblast is a syncytium. It's a structure which is developing by fusion of cells. By fusion of cells. Please, not connection. Fusion of cells. A next nice example in our body, in a human body, is a muscle fiber of a cross-striated skeletal muscle. It's also a syncytium. Fusion of cells. So in this moment, the fusion of cells when this is syncytiotrophoblast, so there are still nicely seen cells, is automatically called the cytotrophoblast. So the fusion of cells of cytotrophoblast, we come to syncytiotrophoblast. And you see, the processes will be longer and bigger, pulling on the whole blastocyst into the endometrium. So this is around the 7th day after fertilization. The implantation of the blastocyst is starting. The 6th day, once more repeating, 7th day. So, syncytiotrophoblast, cytotrophoblast. Later, blastocyst. Embryoblast, cavity, trophoblast, embryoblast. Let's leave for a few minutes the blastocyst. Let's concentrate on the endometrium. Because something happens also here, in the endometrium of the uterus. So, and what happens here? The decidual reaction. The endometrium, in the moment the blastocyst wants to implantate inside, is in a perfect secretory phase of the menstrual cycle. You see here the endometrium, so high. In a human, 7 to 8 millimeters. 7 to 8 millimeters. The next layer is a muscle layer of the wall of the uterus, and this is the smooth muscle cells myometrium. The blastocyst has to move inside the endometrium, please. Completely inside, by implantation. You see a nice, perfect picture, histological slide, a perfectly prepared secretory phase of the menstrual cycle in the uterus. This secretory phase of the menstrual cycle will happen under the influence of progesterone. Progesterone is a steroid hormone produced in the yellow body, corpus luteum in the ovary. Graafian follicle after ovulation is developing into a yellow body, corpus luteum, and the cells in corpus luteum are starting to produce progesterone, and he has got the biggest influence to change the proliferative phase, which is before ovulation, into this, what you see now, secretory phase, after ovulation. And now, when the blastocyst is starting to move inside implantation, this endometrium in the secretory phase will undergo a perfect right decidual reaction. This decidual reaction is involved mainly in fibroblasts, mainly running in fibroblasts. Fibroblast is a basic important cell in a loose connective tissue, or let's say generally in a connective tissue, with a oval-shaped nucleus, nucleolus, elongated spindle-shaped cell with short processes, with a basophilic cytoplasm, rough endoplasmic reticulum, Golgi apparatus. A fibroblast is producing the basic molecules of the extracellular matrix. That means in the endometrium are many fibroblasts everywhere. And the decidual erection is a reaction which is running in fibroblasts. They are stopping to produce the extracellular matrix, not important in this moment now. The cells has to change the shape into decidual cells, storing glycogen, storing glycoproteins, storing glycolipids. Nutrition for the cells of the implantating blastocyst. Decidual cells, former fibroblasts. And now we have 4 basic types of decidua according to thanks to localization of the blastocyst in the endometrium. Above here would be the myometrium, the muscle layer. Between the blastocyst and myometrium is decidua basalis. So here the reaction will be mostly intensive, nice. You will see nicest, perfect, big decidual cells. Why at this place? Why at this place between the blastocyst and the myometrium? Why here? Because decidua basalis is a very important part for the development of the placenta later. Around the blastocyst is decidua marginalis, also running a decidual erection. At this place, you see through this hole came inside the blastocyst. This will be once more covered by an epithelium, by connective tissue, and here the reaction is very weak, decidua capsularis. We don't need the placenta here, we need the placenta on this side. The rest of the endometrium will undergo the reaction very weakly, and this is called decidua parietalis. So the most nicest decidual cells we see in decidua basalis. Here is the reaction the biggest. So I've drawn you a picture of a fibroblast, and now how look the decidual cells. So you see that the former fibroblast has changed into oval-shaped, round, spherical, big, large cells in the endometrium. Nucleus, you see still, but it's a round nucleus, not anymore elongated, oval-shaped, but a round spherical nucleus. In the cytoplasm, many lipid droplets. You see around the decidual cells a blue color of the extracellular matrix with glycosaminoglycans. They are also polysaccharides. This staining is Alcian blue, counterstained with nuclear red. So this is a perfect decidua. Decidua is from a Latin name, decidus, which means to fall away, decidus. And those cells will give the first nutrition for the implantating blastocyst, these decidual cells, which is called histiotrophy. Later, when the nutrition will be given to the cells of the blastocyst by the maternal blood, this is called hemotrophy. But on the beginning, the first days, the nutrition for the cells is from decidual cells, former fibroblasts, and this type of nutrition is called here histiotrophy. So I hope you understood that you see here the epithelium of the endometrium. Here is a gland in the uterus. You see that here would be running the decidual reaction. Here will be decidua basalis. I think you understand it now. And you see the implantating blastocyst into the endometrium. You see that it's around, still opened here, the whole big. So it's around the 8th, maybe 9th day. Let's come back from, this is a syncytiotrophoblast, this is a cytotrophoblast, and now what happens in the embryoblast. This is a cavity of the blastocyst, I think you know these names, and now what happens in the embryoblast. Two interesting procedures. First, by delamination, we see here on the bottom of the embryoblast such not columnar but cuboidal, slightly squamous cells by delamination. This procedure, how they happen to be here, is delamination, and they see the cells of hypoblast. It's interesting that many books and many textbooks and many embryologists, you will see in your books, is using automatically yellow color to show you hypoblast cells. Okay, why not? The next procedures in the rest of embryoblast. First is, where is it written? So, something happens by dehiscentio, that means to push away the cells from one another, dehiscentio, Latin name, dehiscentio. And the second procedure which happens is slightly apoptosis, a programmed cell death. And thanks to these two procedures, this cavity will be bigger and bigger and bigger, and we have here the amniotic cavity, amniotic cavity, amniotic cavity. To the cells of hypoblast, you will suddenly see very high columnar cells, the cells of epiblast. The cells on the opposite side, which has a cuboidal shape or polyhedral shape, forming the ceiling. This is the bottom of the amniotic cavity, epiblast, and the ceiling of the amniotic cavity is from amnioblasts. So the amniotic cavity is surrounded by amnioblasts and epiblast cells. So this is around the 8th, 9th day after fertilization. First, let's move once more to Decidua basalis. I hope you already understand where will be the biggest and nicest decidual cells, Decidua basalis. You see cytotrophoblast, syncytiotrophoblast, and in those lacunae is the maternal blood. From this moment, the cells are getting nutrition already from the blood of the mother. So you see the beginning of hemotrophy for the cells of the blastocyst. You see hypoblast cells. You see epiblast. You see amniotic cavity and amnioblasts. What is new on this picture? In this moment, we see in this former cavity of blastocyst new cells. Very small, irregular star-shaped cells which are starting to fill the cavity of the blastocyst. Irregular, small, star-shaped cells with very short processes. And where is the origin of those cells? Former older embryology books, textbooks, 40 years ago, 60 years ago, were explaining that those cells, red cells, which we call the primary extraembryonic mesoderm, primary extraembryonic mesoderm, the origin is from cytotrophoblast. This is not true anymore. Today we know that those red cells primary extraembryonic mesoderm, the origin is from hypoblast and later from epiblast. Those two lists, so these two types of cells are giving origin to primary extraembryonic mesoderm. So suddenly you see here such a new population of interesting star-shaped cells, but some of them get very flat, you see even the nucleus, surrounding such a smaller cavity in this moment, which cannot be anymore the cavity of the blastocyst, not anymore. But it's so called the primary umbilical vesicle or primary yolk sac. Primary yolk sac. By what is surrounded the primary yolk sac? At this moment, by the cells of hypoblast, and the rest by a very interesting membrane formed from very flat cells, and this membrane has described Mr. Heuser. So it's called the Heuser's membrane. The primary yolk sac is surrounded by hypoblast cells and Heuser's membrane, in another name, the exocellic membrane. Primary extraembryonic mesoderm, those cells are from hypoblast and epiblast. Not anymore from cytotrophoblast, how it was meant before. We are a little bit later. Why? It could be around the 14th day, because you see the epithelium is back over the hole which was running inside the blastocyst. Even you see a small amount of Decidua capsularis. Of course here Decidua basalis on the opposite side. Amniotic cavity, understand, hypoblast, epiblast. Primary yolk sac was a bigger cavity. But what happens now? Division of hypoblast cells, and they are moving on the Heuser's membrane. This is the first important thing. Second, cavities in this primary extraembryonic mesoderm are starting to fuse and to be bigger of the extraembryonic cellum, and they push the extraembryonic mesoderm out and in these two directions. That means how the cavities are bigger and bigger. The primary yolk sac will be smaller and smaller and smaller, and will be, this remained, as a yolk cyst. Yolk cyst. In some books you can find exocellular cyst. It will completely vanish. And these fused cavities will form a big cavity, which is called chorionic cavity or extraembryonic cellum. Extraembryonic cellum or chorionic cavity. In this moment, when you see hypoblast cells in later endoderm of the yolk sac, so you have the secondary yolk sac, not anymore the primary, secondary yolk sac. So you see here how the secondary yolk sac is slowly developing. Here you see the amniotic cavity, amnioblast, and so on. So, this is a cyst which will vanish. This is the remembrance on the primary yolk sac. This stalk will vanish, and this structure will be now surrounded by the large chorionic cavity extraembryonic cellum. Amniotic cavity, here epiblast, hypoblast, secondary yolk sac. Connecting stalk is a connection between the embryo and the outer fetal membrane chorion. This is connecting it. Outer fetal membrane is chorion. How we come to chorion? Primary extraembryonic mesoderm is pushed to cytotrophoblast. From outside is syncytiotrophoblast. 1, 2, 3 layers are forming the outer fetal membrane chorion. Primary extraembryonic mesoderm, cytotrophoblast, syncytiotrophoblast. The inner fetal membrane will be amnion. This will be this. This will be the inner fetal membrane amnion. You see that we have an embryonic disc from two types of cells, hypoblast and epiblast cells. On the end of the second week, we have a simple embryonic disc from two types of cells, the yellow hypoblast cells, the blue higher columnar epiblast cells. This is the result on the end of the second week. Extraembryonic somatopleura, extraembryonic splanchopleura. The primary extraembryonic mesoderm, which is attached to the wall of the yolk sac, is a splanchopleuric. Splanchopleura. This mesoderm attached to the amnioblasts, and here everywhere, is the extraembryonic somatopleura. Extraembryonic somatic mesoderm. And we are on the end of the second week, my dear friends. So, this is the most important thing to say. On the end of the second week, embryonic disc has two layers, hypoblast and epiblast cells. Operculum, the whole, will be covered by epithelium, by connective tissue. The implantation is complete. Blastocyst is inside the endometrium. I explained to you what the chorionic cavity, extraembryonic cell, secondary yolk sac is developing. You know that outer fetal membrane is chorion, this, primary extraembryonic mesoderm, cytotrophoblast, syncytiotrophoblast. Inner fetal membrane, amnion, will be only this. And syncytiotrophoblast, our famous syncytiotrophoblast, which is here the dark green syncytiotrophoblast, from the 7th day is starting to produce a human chorionic gonadotropin. This level of this hormone will increase each day, higher and higher level, because syncytiotrophoblast is developing to be bigger and bigger and bigger. Then this hormone gets into the maternal blood, by maternal blood to the kidneys, in the kidneys into the urine, and 14 days, after 2 weeks after fertilization, if you prove this hormone in your urine, the lady knows definitely that she is now pregnant. Because normally in your body is not present syncytiotrophoblast. Only when it's preparing the way for the blastocyst into your endometrium. Then you can have only this hormone in your body. A nice, perfect slide, photograph of a slide. I think that you understand that here is running decidua reaction intensively. Here will be decidal cells, decidua basalis, between the blastocyst and the myometrium, which is somewhere here. This is a gland in the uterus, in the endometrium. This is a surface epithelium, simple columnar, which is on the surface of the endometrium. This will be decidua marginalis. This is syncytiotrophoblast, this is cytotrophoblast. You see the primary yolk sac and you see the Heuser's membrane. You see the cells of hypoblast. You see the amniotic cavity and the high columnar epiblast cells. Here are amnioblasts. You see the cells of primary extraembryonic mesoderm. So we are around the 12th day after fertilization. The blastocyst will be completely hidden in the endometrium around 2 days later. And the epithelium will grow over this fibrin plaque. This is the same picture as before, higher magnification. Hypoblast, epiblast. Amniotic cavity is very thin here. Those are amnioblasts. Cytotrophoblast, syncytiotrophoblast. Nicely seen here. So this is a higher magnification from this bilaminar embryonic disc. Bilaminar embryonic disc. Yes. Thank you. And that's all from our first lecture from embryology, so-called Embryology 1. And now the next lecture will go on from the 3rd week with the gastrulation and notogenesis.